some things. But on Monday, we were talking about proteins, right? We were talking about protein structure specifically, right? We had walked through those 4 levels, correct? I'm not like misremembering my days. I give these lectures enough and I can't remember where I'm at. And so I think we had kind of talked about motif and domain, but I'm going to go ahead and back up and kind of go through it again. Is that true? We had actually gotten past this slide, I think. I think so. All right, so we were around here, right? Because we had talked about, right, primary, secondary, right? So the chain of amino acids, right? When the chain of amino acids makes those coils and those sheets, that's secondary structure. Those coils and sheets come together, that's tertiary structure. There's something on this. Anyway, and then quaternary structure is when multiple amino acid chains or polypeptides come together, right, and form this giant functional protein, right? So we got through that. And then we were talking about how, right, so the structure of a protein is determined by the amino acid sequence, right? We make this chain of amino acids, they start interacting and folding up and we get a protein. Now those amino acids are coded for by your DNA, right? That is literally what your DNA codes for. Your DNA codes for amino acids, which makes proteins, which then makes you, okay? So with this next generation sequencing, again, this we'll pick that apart like in unit 4 or 5 or something. But suffice to say that we have technology now that can sequence loads and loads and loads of DNA, right? And we can create these huge databases of nucleotide sequences, which are basically the code or the instructions for the amino acids that make proteins. So when you have this kind of big data, you can start looking for patterns, right? In the way that proteins are formed and what kind of amino acid chains make what kind of proteins. And so in looking at these patterns, we've actually kind of added in 2 additional terms for protein structure, okay? The motif and the domain. And motif and domain kind of fall between secondary and tertiary structure, right? So secondary structure, helices and sheets, tertiary structure, our final protein. And so we've got a couple little sub-levels that kind of fall between. All right, so a motif, kind of refer to it as a super secondary structure, okay? So it's a little more than just the coils and the sheets, right? When they started looking at all this data, they realized there are these repeating kind of motifs or patterns in protein sequence. So you frequently see like a beta-beta motif. So you see 2 sheets together that kind of fold together. Maybe you see 2 helices, right? A helice with one of these little kind of elbow bends, right? A helix turned helix, you might say, right? Not that you need to know all these different motifs, but that you see these common patterns in the arrangement of the secondary structure. So common patterns in the arrangement of coils and sheets, basically, right? We call those motifs. Now, slightly larger than a motif, but smaller than our complete protein is a domain, right? So it's kind of a subset of tertiary structure. So we have tertiary structure, right? This whole protein, right? And what we're noticing is that there's these kind of patterns in the region. So you have a series of motifs, say, that come together to form this functional domain. So you've got this whole protein and different regions will do different things. So in this particular example, this kind of lavender colored region of the protein is specifically for anchoring the protein in a cell membrane, right? So you get this functional domain, this region of a protein that is for doing some specific thing, right? So this is a membrane protein that's pushing stuff in and out of a cell, probably so. We're not going to worry about what the other parts is. It's just an example, right? So basically you have a whole protein and you've got different domains of a protein that are doing different jobs for that protein, right? And so then a whole set of domains are going to make up your full protein, that tertiary structure. All right. Now, if we're making these giant complicated proteins out of hundreds and hundreds of amino acids and then folding multiple polypeptide chains together to make proteins. And it is critical that we fold the proteins correctly, right? For a protein to function, it must take a very specific shape. And so something recent-ish that we have found is that some protein folding, especially in these big complicated proteins, will rely on these other proteins called chaperone proteins, right? And they literally do that. So you've got a protein and the chaperone proteins literally kind of chaperone that protein, make sure it comes into the shape it's supposed to, right? So they help other proteins to fold correctly. You see this in one example, heat shock proteins. So proteins, like any compound, like any molecule, if you heat them up, they fall apart, right? Like they melt, right? Literally, at kind of like the largest scale. But even if they don't melt, if you heat up a protein, it starts to lose that secondary structure. Those hydrogen bonds kind of start to let go, right? So you heat a protein, you lose those sheets and coils, and we say the protein denatures, right? When you melt butter or when you fry an egg, right? You're causing the proteins to kind of lose whatever conformation they're supposed to have, right? And a heat shock protein deals specifically with that. So proteins that have denatured, that have gotten too warm and kind of gone a little loosey-goosey, you have heat shock proteins that kind of fix them. We'll kind of pull them back together. I mean, once the egg is fried, you're a bit beyond a heat shock protein's help. It's probably also fried the heat shock protein, but kind of within the range of things happening in your body, right? You've got these heat shock proteins that help proteins recover from getting a bit too warm, all right? And so deficiencies in these chaperone proteins can result in disease, right? Which makes sense. You've got proteins that you must have that must be the right shape to do things. And so if you have some deficiency in your chaperone proteins, then you might have proteins that don't take the form they're supposed to, don't serve their purpose they're supposed to. For example, cystic fibrosis is this kind of a disease. So you end up with these incorrectly folded proteins that are supposed to be moving ions across cell membranes, ultimately working in the formation of mucus, right? Because your gut, your lungs, everything in your body basically is lined with mucus, which honestly kind of grosses me out to talk about a little bit. But if these ions, right, if this protein's not functioning properly, all your mucosal linings, the mucus gets too thick. And so you end up with respiratory and digestive and all kinds of issues because your body is not lined properly, basically at that point. But it's all because you're lacking these chaperone proteins that are supposed to be keeping things folded up the way they're supposed to, right? So this idea of denaturation. So like we talked about, heat is one thing that will cause proteins to kind of lose their shape a little bit, but denaturation can happen for multiple reasons, right? Proteins can change shape or unfold in response to lots of environmental factors. So temperature is one. Things like pH, it becomes very acidic, right? Which your body shouldn't, right? That's that whole thing where your body needs to stay pretty neutral. Ion concentration, right? Which kind of makes sense, right? The protein is folding based on all the different charges in those amino acids. And if you put that protein in a very like ionically charged environment, it's going to change the shape that protein takes. So these changes in shapes are called denaturing, right? And this is what pickling is. When you put vegetables or eggs or whatever in vinegar in a really, really acidic environment, you're denaturing the proteins and subsequently you're preserving these foods, right? If you add loads of salt, i.e. changing the ion concentration, right? You're denaturing the proteins in this food, which is different from dissociation. So occasionally, right? So denaturation is a change in secondary structure that kind of causes things to unfold a bit. But we can also have quaternary structure that comes apart, right? So secondary and tertiary structure is just kind of unfolding, but that quaternary structure where multiple peptides stuck together. And if those multiple polypeptides become unstuck, right, they've dissociated, right? The pieces of the functional protein have come apart, right? So just kind of differentiate those two ideas. So denaturation is just kind of a loosening of the secondary and tertiary structure, right? But if we actually start pulling the subunits apart at quaternary structure, that's dissociation. Okay. Which I believe, yes, finishes proteins for us. Proteins are done. Last one, we've got lipids. All right. So lipids, another one that we're vaguely familiar with in different forms in the nutritional sense anyway. So we'll talk about, we're going to use triglycerides as our kind of main lipid molecule example. Lipids are going to be a little different than these other polymers we've been talking about. We'll talk about why, talk about function of fats, and then also talk about another function. So mostly is energy storage, but then another function is the formation of membranes, like cell membranes. And so that's kind of where we'll end unit 3. So lipids are insoluble in water. That's important, right? If they're for energy storage, you don't want something that's just going to kind of like fall apart in the system. So they're insoluble in water. They have a high proportion of carbon-hydrogen bonds. They are mainly made up of carbon-hydrogen bonds, hence being good energy storage molecules. They can't fold like a protein, right? So they're not going to take any special conformation. They're just going to kind of be these long hydrocarbon holding molecules, right? And in water, right, they're going to have these hydrophobic regions, right? Hydrocarbons are nonpolar, so they're hydrophobic. And so in water, they're going to cluster together, right? That idea of hydrophobic exclusion that we talked about when we were talking about properties of water, right? Water doesn't like nonpolar things. It's going to push them away. And so subsequently, the lipids in your body, the nonpolar things are going to kind of gather together, right? Similar to oil droplets or things like that. And this is where we see the formation of cell membranes. All right, so when we talked about sugars, carbohydrates, right? They're made of monomers, monosaccharides, into polymers, polysaccharides. When we talked about nucleic acids, we had monomers, nucleotides, making nucleic acids are polymers, right? And then in proteins, we had amino acids making polypeptides. Lipids are different in that they don't have that kind of easy monomer polymer thing going on. What we end up with are two basic types of molecules. We're going to keep, there's lots of kinds of fats. We're going to keep it fairly simple. We're going to have a handful of fatty acids attached to a glycerol, and that's going to be the end of it. We're not going to make these huge long chains of monomers like we've been making with the other molecules, right? So a fatty acid is basically a long hydrocarbon chain, longer than this, right? A long hydrocarbon chain with a carboxylic acid at one end. So lots of carbons, all full of hydrogen, and then a carbon double bonded O and a hydroxyl on one end. All right, so we've got these hydrocarbon chains, and we're going to attach those to a glycerol. And a glycerol is just this little 3-carbon polyalcohol. So 3 carbons with 3 hydroxyls, and then we just fill the rest out with hydrogen, right? So we've got these glycerols, we've got these fatty acids. We're going to put them together the same way, dehydration reaction or hydrolysis, right? So I'm going to take this fatty acid, we're going to lose water, right? A hydroxyl and a hydrogen. And we're going to attach that fatty acid to one of those carbons, right? So basically just like we've done everything else up to this point. But you notice we've got 2 more parking spots left, right? So we're going to make a triglyceride. We're going to plug 3 fatty acids into this glycerol. You don't need to draw all these. It's going to get quite chunky, right? We're going to put 2 more in there. It gets real messy. So let's just calm down a little bit. Triglycerides. So we've got a glycerol We do 3 dehydration reactions to attach 3 of these fatty acids, these hydrocarbon chains to it, triglyceride. This will be, if you've been in lab, you probably saw these guys already, right? It's kind of our basic lipid example. If you've got lab today, then you get kind of like a bonus review session. Hopefully it's helpful. All right, now we've got this triglyceride. And you've probably, if you're kind of into nutrition at all, heard lots of words around lipids, right? We talk about fats being saturated or unsaturated, right? We talk about trans fats and all those sorts of things. And all of those words are actually relating to the molecular structure of the fat. So if we say a fat is saturated, it means that in these fatty acids, right, these long carbon chains, that it is saturated with hydrogen, right? All our carbons are single bonded and we have as many hydrogens attached as will fit. Saturated fat is saturated with hydrogen. This is most animal fats, right? Because if you just have this long chain and it's all full of hydrogen, you get these very linear kind of molecules. So they can stack really close together. And so chemically what that means, if you have these aligned molecules, right, that are very close together, it increases, right? It's got a higher melting point. So saturated fats, animal fats are solid at room temperature. If you eat meat, you think about fat on meat, right? It's solid at room temperature. Butter is solid at room temperature, right? Versus unsaturated fats, where we have some double bonds in our carbons. And when you have a double bond in your carbon, right, we lose 2 hydrogen, right? So we've lost 2 hydrogen, right? And so we call that unsaturated. It is not saturated with hydrogen. And what this will cause to happen, right, because these 2 hydrogen are missing, the chain's going to kind of bend, right? You're going to get like a little kink in the chain here, right? So this is most plant fats. The molecules are bending at those double bonds, so they don't lay all nice and tidy together. They're more kind of spaced out and loose, which causes these to be liquid at room temperature, like olive oil or vegetable oil, things like that, right? They're liquid because they're unsaturated. To get a little more descriptive, right? If there's only one double bond in the chain, it's mono-unsaturated, right? It's unsaturated at one spot. If there's multiple double bonds, right, it's polyunsaturated. It's unsaturated at multiple spots in the fat. Right, so when you see those labels on food, this is what it's talking about. Now in these unsaturated fats, right, this is where trans fats happen. So if you've got these double bonded carbons, naturally, right, like in plants, you're missing the hydrogens off the same side and they kind of tip together, right? You just get these little kind of bends, right? We call this cis configuration because the hydrogens are on the same side. Versus a trans configuration, right? If you do this, if you industrially hydrogenate your oils, right? You have these hydrogenated oils, you end up with a trans fat where the hydrogens are pointing in opposite directions off either side of the double bond. And instead of having a little bend, you get kind of a little like slope, right? This increases the shelf life of the fats, right? This is margarine, right? But they're not really great for consumption, right? These are no longer, right? These industrially created fats are no longer considered safe by the Food and Drug Administration, right? So we tend to try to avoid them, right? So you can have unsaturated fats, naturally occurring unsaturated fats that are in this kind of cis formation versus trans fats that are created industrially. All right, now, kind of all we're going to do with lipid structure. Like I said, there's a lot of kinds of fats. This is the only one we're going to kind of pull apart too much until we get to membranes. But there are lots of other types of fats, just examples of other things that lipids are doing. Terpenes, are lipids. So these are components of pigments and things like chlorophyll. Terpenes are also what's harvested if you're looking at like natural rubbers. Terpenes are lipids that are harvested for natural rubber from trees. And steroids, which is one that I think doesn't kind of naturally come to mind. Steroids are actually lipids. So cholesterol, estrogen, testosterone are all lipids as well. All right, we're not going to worry about them structurally, just to know that lipids can do a few different things. All right. Now, lipids have one last function that's really important. It's going to kind of carry us into Unit 2. Unit 2 is all about kind of cell structure and function, right? And so a big part of that, oh, we got one more, right? Energy, sorry. Energetics and fats, right? So most of these fats contain more than 40 carbon atoms, which is double the ratio, right, of carbon-hydrogen bonds that you see in things like sugars and carbohydrates, right? So they basically are storing, you don't need to know how many specifically, but more than twice as much energy per gram as you get in a sugar and fats, right? And since they're insoluble, they're really stable. They can hold lots of energy. So this is right, this is your body's long-term storage. So if you consume excess carbohydrates, your body's going to store that as starch if you're a plant, glycogen or fat, right? It's not a bad thing as long as you use it eventually, right? But diets heavy in fat, right, has some kind of health implications, right? It's associated with heart disease, among other things. Atherosclerosis, this hardening or lining of the arteries is correlated with this kind of excessive fat intake in the diet. So it's something to be mindful of, right, just nutritionally. But they're still important. You do still need fat on your body, right? Everybody's got some amount of fat on their body for storing energy for various things. Now the last and final function of lipids, which will be the last and final thing for chapter 3 and unit 1, are that lipids are also the main components of membranes, of cell membranes, right? The phospholipid bilayer, if you've done any cell stuff before, right, is obviously made of lipids. Specifically, different than triglycerides, membranes are made of phospholipids, all right? And so phospholipids are made up of glycerol again, right? Half of this looks very similar to what we've seen before, right? We've got a glycerol with 2 fatty acids attached, but instead of a third fatty acid, we're going to attach a phosphate, hence the phospholipid, a phosphate and some organic molecule, right? The key thing about this organic molecule is it's going to be charged, right? So now we've got these hydrocarbons, nonpolar, hydrophobic, and on this end, we've tacked a charge molecule. So now we're hydrophilic on this end because we've got a charge. Water's going to like that. Water's not going to like this, right? It's going to push away those fatty acid tails. So these phospholipids, glycerol with fatty acids, and then a phosphate and a charged molecule on the other side. So we tend to represent these kind of like this, right? This polar head is the phosphate group and charged molecule, and then these non-polar tails, right? Those fatty acid chains. So we've got a polar region and a non-polar region. So when these are in water, they're going to behave in a very specific way, which we've talked about before. Right? So in water, the nonpolar tails are going to aggregate, right? Because of hydrophobic exclusion. Water doesn't want to interact with nonpolar stuff. It's going to push it away. They're all going to kind of get shoved together, right? And then on the outside, you're going to have the polar heads, right? Because they're going to be interacting with the water. So certain phospholipids will just form these micelles, right, which are just these kind of balls of lipids where the nonpolar part's in the center and the polar part's on the outside. Now, this isn't a cell membrane, right? This is just kind of like those little oil globs that we've seen before. There are certain phospholipids that line up slightly differently, right, so that they make these sheets, right? So polar head, nonpolar tails. And in the case of these phospholipids, they form a bilayer. So you just imagine this being kind of a big sheet of these phospholipid molecules, right, that just kind of comes out. So the water pushes the hydrophobic tails inward. The hydrophilic heads, right, are outside where the water is. And so these lipid bilayers then, hydrophilic on the outside, hydrophobic on the inside, become the main component of cell membranes, right, biological membranes, right, that we'll be talking about when we get into chapter 4 and talking about cell structure and function. All right. But for our unit 1 purposes, we know that lipids are for energy storage. They're found in things like chlorophyll and pigments and steroids, and that because of these hydrophobic and hydrophilic properties will form membranes in water, which is important. Okay. So our lipids as membranes. All right. All right, hold on. There's a typo on this slide and it's been bugging me all morning. I'm fixing it. There we go, on this slide. So that is the end of chapter 3. That is the end of material for Friday's exam. All right, so we have a little time. So we've got our kind of end of chapter wrap up review questions just to kind of remind ourselves what we've been talking about. So, right, so one thing that came up with all of these molecules were dehydration and hydrolysis reactions. What's the difference between dehydration and hydrolysis reactions? What are they for? What happens? What do they do? Any of these? What happens in a dehydration reaction? Yeah, right? That's how we turn monomers into polymers, by losing, what do we take out? What's lost in a dehydration reaction?
OH.
If you're dehydrating something, what are you losing? Yeah, you lose water, right? And that's how a lot of these polymers, basically all of these molecules that we're talking about, right, the pieces come together through dehydration reaction. So what's a hydrolysis reaction? Yeah, you add water. And what happens when you add water in these reactions? Yeah, you break those pieces apart, right? So you can remove water and attach them together, or you can add water, right, and break them apart. Good. Primary function of carbohydrates?
Energy
storage, right? Right, those are sugars. They store energy. What are the monomers of a nucleic acid? Nucleic acids are long chains of what? Is it what? Not pentose. That was one of the monomers for the saccharides, right? For the sugars. Nucleic acids are like DNA. What were the monomers for DNA?
Deoxide root.
So DNA is, oh no, I'm going to say it wrong. Deoxyribonucleic acid. And it is made of chains of monomers of, there's 4 of them. Well, there's 5 of them, technically. Not starches. Starches are carbohydrates. Yeah, adenine, guanine, cytosine, thymine, uracil are all what are they? It also starts with an N. Nucleotides, yep. What are the monomers of proteins? What are the units we're attaching together in proteins? Proteins are made of long chains of amino acids. Yep. All right, so we talked about so much. We talked so much about protein organization. There are 4 main levels, right? Primary structure was the the what? The sequence of the sequence of amino acids, right? One level above that is secondary structure. So what are we forming in secondary structure? Hydrogen bonds, which cause the amino acid chain to form what's?
Peptide groups.
Not peptide groups. They form a couple different shapes. Helix or coils and, what was the other one? Sheets, coils and sheets, right? And so then tertiary structure is what? Yep, that's the final complete protein. And if a protein has quaternary structure, what are we looking at then? Yep, we've got multiple polypeptide chains in their tertiary structure and we plug them together. Right, that's our quaternary structure. All right, why do lipid bilayers form? Or maybe why do certain phospholipids form bilayers? If that's a better worded question for you. That's the last thing we talked about, right?
The framework of biological membranes.
Yeah, yes, it came in twice. Yeah, they form biological membranes. Why? What about them causes them to form these membranes? Yeah, right. The water pushes all the nonpolar parts together and leaves the polar parts on the outside. What is that called? Right, it's one of those characteristics of water to push.
Hydrophilic.
The other one, hydrophobic what? Pushes them away, excludes them, one might say.
And they're
nonpolar, they're hydrophobic. Water excludes hydrophobic things, and that is called?
Dehydration.
No. It's just those words that I said, which is kind of like in the other order. Water excludes hydrophobic things, and that is called? Water excluding hydrophobic things is called hydrophobic.
Are you talking about hydrophobic exclusion?
Yeah, hydrophobic exclusion. That's it. That was all. Yeah. Bilayers of lipids form because of hydrophobic exclusion. Nonpolar stuff comes together, polar stuff keeps interacting with the water, and that gets bilayers. All right. Any other questions? Chapters 1 through 3, concerns about the exam? Other feelings about things?
How many questions is it?